Hi, Sarah, you're live.
Good day, ladies and gentlemen. Welcome to session 1 on ecology. I'm Sarel Celia from Northwest University in Potchefstroom. We invite you to the first 4 presentations of this session. If you have any questions, please put the questions in the Q&A box. The chat box is for any comments or but not for questions. So without any further ado, I would like to introduce our first presenter, is Prof. Marion Meyer, and he will tell us more about fairy circles. Over to you, Marion.
Thank you very much. Thank you very much for everyone for joining. I appreciate that. Do you see my presentation? Can I continue? I think should see my presentation now. So I'm going to start, and stop me if you don't see it. During the last number of years that we've been working in the Namibian desert on the fairy circles, we noticed often that some of the big succulent Euphorbia species are dying in big numbers, Gumifera gregaria, as well as Demarana. We then decided to determine if we can quantify the state of health of these Euphorbias, if our observations are correct or not. So just a quick background, the fairy circles are located on the coastline area, mostly of Namibia. We found them a year or 2 ago in this area and even up into the Kalahari Desert. Their distribution matched the distribution of Euphorbia gregaria very well in this area, Gumifera very well in that area, and then in the north, that of Damarana. In short, the main theories on the formation of fairy circles are gas, termites, self-organization, and then last year we provided some evidence that Euphorbias caused them. We saw in many places dead Euphorbias that's been there for decades, according to previous reports, and all of them you will still not see them being covered in grass. We've obtained an aerial photograph of the Garoep region in southern Namibia. This is a small part of the aerial photograph. It was taken in 1966. We ground truth all these black spots and found them to be Euphorbia gumifera. There are no other big species growing in that environment. We then determined what happened to these Euphorbia gumiferas in 2015, ground truthed them all, of course, found that 134 of them have died and formed fairy circles, or what looks like the beginning of fairy circles. Another 69, that is now of the 406, were also dead, not yet forming fairy circles, they were busy decaying. So shockingly to us, that is half of the species that were on the aerial photograph are dead. We looked for seedlings or young plants, found virtually none. The same applies to Euphorbia gregaria near the Fish River Canyon. If you have sandy deposits, you find many dead Euphorbia gregarias. So if you would do historical imaging comparison, we have also seen that several of them are busy dying or either already dead a 10-year period roughly. So our hypothesis is that succulent Euphorbias are declining in high numbers on sandy soil because possibly of climate change and the low water capacity of sand. I flew with a micro ride over the Brandberg area, and it was clear to see that they are performing very well. They look very healthy on hills and on mountains. When you get to sandy areas, they have basically disappeared, or you will see them in some places, but they will survive on Inselbergs or koppies or hills. If you would zoom into many of the sandy deposits from southern to northern Namibia, you will often see this site of a few remaining Euphorbia damaranas in the north and then Gumifera or gregarias in the south. Louis Scott from the University of Free State did some very interesting work on fossilized Hyrax middens. He did this work in the Sandsea area. I'll show you a map just now. But he found that the Euphorbia pollen, if you look and see the red parts of these bars, were a substantial percentage of the pollen of the big species or tree species in the area where we work. Then it seemed to get much less and stayed just a few percentage points of the big species. So Louis Scott collected the Hyrax middens in this area, Namib Sandsea. In the Namib Sandsea, between the dunes, you will often see thousands of large fairy circles. Now the question is then, where are the Euphorbias? In this region, there are no Euphorbias left over. If you look, you will find them about 100 kilometers away from this area. So how do we explain that? I'm not sure yet, but it could be coincidence or not. But during the time that the pollen decreased substantially, 1,300 years before present approximately, that was one of the driest periods that Namibia has experienced in the last 2,500 years. These records were from the Spitskoppe, just north of Brandberg, or south of Brandberg, very close by. If you look at temperature records, then at about 1,200, 300 years ago, it was very warm. So it is a possibility that they died out on sandy areas, and that is the key word, because the stress is so much more if you have drier, warmer conditions on sand. So the aim of our study then is to compare the state of health and quantify that of Euphorbia damarana plants by comparing satellite images of 2012 and 2021, and then determining the NDVI values. The NDVI values is a very good measurement of photosynthetic activity or the presence of chlorophyll, in other words, alive plants. I just want to mention as well that I focus on this Euphorbia damarana and this talk because of our previous work on the fairy circles, but we saw many other species that didn't look good on sandy deposits. So what is the NDVI values that you can determine? If you take a healthy plant, it will absorb a lot of red light for its photosynthetic activity and reflect a lot of near infrared because of the chlorophyll. It will reflect less red light back because it's used in photosynthesis. If you have a stressed plant, the red light reflection will be more, relatively more, because there are no or very little photosynthetic activity. If you then look for at a near infrared light, less will be reflected back because of chlorophyll breakdown. A plant with an NDVI value of roughly 0.33 would be stressed or diseased. We identified 2 sandy analysis sites and 2 Inselbergs or hills surrounded by sand in the Brandberg area on satellite images. Then identified 40 Euphorbia damarana plants per site on satellite images. So we had to go there and ground truth that what we saw on satellite images were actually Euphorbia damarana. So we confirmed that and we could proceed. So the healthy and dead Euphorbia damarana plants were digitized as polygons, so a little circle or whatever was drawn around them. And we did that with QGIS. We then used these polygon shape files with the Euphorbia being marked, lifted that from the raster or that map or that image, and we could determine the NDVI values of the Euphorbias themselves by using a raster calculation algorithm. The results were then extracted from QGIS and analyzed in Excel with the t-test. This is what the area looks like. Very sandy. There is Brandberg, Oegap River running through there. Thousands and thousands of fairy circles. Here's some Euphorbia damaranas remaining in some areas there. This is where Brandberg is situated. The sites were 500 by 500 meters each. Here you see a false color image of a small part of one of the analysis sites. Here in Damarana 1.2, you will see a healthy Damarana plant. The false color gives you the near infrared reflection or focuses on that. That is your band 1 if you do this type of analysis. If it's not a bright pinkish color or blackish like that one, then it's dead. Here we can even see remains of a few Euphorbia damaranas. These were ground truth, so we were there. And then you will see some in-betweens. That is sort of brownish, pinkish, and that would be like 50–60% dead. So our results then were that we saw many dead Euphorbias on the sandy deposits, even like this. Big one is dead, that one is partially dead. And on the hills and the mountains, they looked lovely and healthy. We, in each side, then looked for seedlings or small plants, and it was shocking to find in these 500 by 500 meter sites, 4 plants smaller than 2 meters inside one, and 1 only inside 2. Here you have a video, short 30 seconds video, to show just some part of this, of the amount of dead. This is the remains of a dead Euphorbia. There's the other remains. There's dead ones. This was now taken in November this year. That's a healthy one, or partially healthy. Here is a 100% healthy one. And here you can see a number of dead ones also. Okay, we can move on. How did we do the Ndvi determination? Here is just an example. In this interesting C-shaped growth form or growth pattern of these Euphorbias, there are some alive, like the top one. In the middle, we also see another one that's alive. And then here we see a number of dead ones. If you would look at the false color of this, you will see the bright or pinkish, purplish colors indicating living plants or partially living plants, and then dead ones and dead ones. Because these were marked, in this example, we marked 6. They were marked, and we could extract them from that raster and do the Ndvi calculation. So the bright red ones for the Ndvi are very healthy, and the ones with a lot of white in them shows there's this it's partially dead or 80% dead in that particular case. So our results then showed that in all places that we analyzed, the Inselbergs, the solid green bars being 2012, striped ones 2021, we could see a significant difference in the Ndvi value. Therefore, much less photosynthesis, less chlorophyll. In each of the sites, we had the same significant P smaller than 0.05 difference. What I need to say as well, we saw many, and we noted that, and we will also report that one day if we publish that. There were many dead, just remains of Euphorbia damaranas. We're doing chemical analysis, metabolomics of these also to be sure that it's damaranas. And they were, of course, not taken into the Ndvi determinations because there's nothing left there. Just then, what happened in the past in that region? So we constructed a polygon that you see in white here around this area, covering our analysis sites. And the rainfall you will see then since 2012 of that area It's wonderful data one can get on Climate Surf, the website of Server Global, data being supplied by NASA. And you can see how small or how little the precipitation was since 2012. You can also determine the NDVI values from this website. We then, in this polygon, it would have taken account of the plants in the Ughab, the Damaranas around here, and basically no grass during this time, but in previous times there were. So we were shocked to see that the NDVI values were this high before 2012. We then analyzed end of 2012 around here and compared it to the current, this is then September also of last year. So if we would have had satellite pictures of images of previous times, the difference would have been much higher. And what we learned from this is that you cannot just pick 2 years and make a good, be sure of what you see on NDVI values. So you have to look at a long-term trend.
Marion, you have 1 minute. You have 1 minute left, Marion.
Thank you very much. 2 slides left. So, like the lonely survivors in the sand. So this is then just a typical photograph from a microlight to show here and there in between the fairy circles, which is most probably the tombstones of previous growth of Euphorbia damarana, and we have in the south the same with Gumifera and Gregaria. Conclusions then, these 3 succulent species are declining in areas of sandy soil, and it is possible, very difficult to determine if climate change is the main driver. It is then proposed that hundreds of thousands, millions, some scientists say, of fairy circles are the tombstones of the earlier presence of succulent Euphorbias, and therefore a startling indication of climate change. We will expand this study to many more sites and locations in future. Thank you very much to Digital Globe for providing a number of satellite images for free, and to many other people that help us in the process. That's it, Sarah. I will stop sharing.
Thank you very much, Marion. It was very, very interesting. Yeah, I don't think those fairy circles will have the same meaning to everybody seeing them in the future. Ladies and gentlemen, I did not receive any Q&As. Are there any questions? If you want to ask a question, you are just welcome to switch your microphone and your camera on. Marion, I've got a question, and I'm not a physiologist at all, but isn't this a very time-consuming process? Aren't there any other quicker, more field-based studies that one can have the same results?
Sorrel, the advantage of this is that you of course, you have to go into the field to ground truth that what you see with Euphorbia, it's easy. They have a typical intense green bluish color. They are basically round. So we know them from work for 10 years. So when you identify them on your images, satellite images, then it becomes very quickly. So you just digitize them and you do a calculation. And if you have learned that, I've done that now during the lockdown while I was in isolation. I was forced to be not on holiday, but here, and I learned how to do those things actually, and it is faster. But you have to be sure that what you see is what there is.
Definitely, yeah, that's important. We've
got
hot. A question.
It's very hot.
We've got a question from Sandy Lynn Steenhuisen. She says, "Thank you, Marion. What are the population number like for those species? Are they classed as endangered?"
No, they will not be classed as endangered. So what I have to stress again is that they look very good and very healthy on clay soil, on hillsides, and in the mountains. But on the sand, you see them in nearly all places where they're on Koppies and Inselbergs, you will see them on sand as well. But in some areas, they're just gone. And we think that is what happened there in the desert sand sea.
Okay, thank you. Dave Berger also have a question. He said, is there any data of the lifespan of these Euphorbias under good conditions?
Thank you, Dave. That's a very good question. So no, we did some radiocarbon dating. We got indications, but the numbers vary between our different samples we gave to the guys from WITS. But they were like 200 years. So we took remains of a few dead branches and things, and those were dated like 200 or 300 years old. So I think it's quite long, but I recently spoke to the scientists from Oxford, and I'm very excited. We're going there next April, or this April, and we have an interesting way of going to go and try and determine the age of fairy circles, but then also the age of the Euphorbias, which we believe caused them.
Thank you, Marion. I think we've just came to the end of our Q&A session. Thank you very much again for a very, very interesting presentation. Thank you.
Thank you very much, Sarah. I appreciate it.
Our next presenter is Emma Ferreira, and she will tell us more about pollination rewards in 3 Nemesia species from Namaqualand. Over to you, Emma.
Okay. Good day all. I'm Emma Ferreira, and today I will be presenting my research on the possible effect of the community composition on pollination rewards of 3 Nemesia species in Namaqualand. So Nemesia is a member of the Scrophulariaceae family, which is often referred to as the Leobeckies or Cape snapdragon. There is roughly 77 species that are endemic to southern and eastern Africa, where they are mostly concentrated in the winter rainfall regions. Nemesia have nectar spurs, which is thought to be a key innovation. A key innovation is a novel trait which is either behavioral or morphological in manner and linked to the adaptability and speciation of taxa. Nectar spurs is thought to be driving diversification in other taxa. However, it is believed that the climate change of southwestern Africa coincides with the radiation of diversification of Nemesia. So the knowledge gaps that we hope to fill revolves around the primary driver of diversification. Was the primary driver pollination specialization or ecological specialization? What is the significance of this research? This research falls under a greater collaborative project concerning the evolutionary diversification of Nemesia. It will contribute to our knowledge on the ecology of Nemesia and to the understanding of the role of nectar spurs in the evolution of Nemesia. The aim of this research was to assess the role of nectar spurs and ecological parameters that may have influenced the diversification of 3 morphologically similar species, Nemesia nisacarpa, Macroceras, and Palchella. So the objectives were to compare the 3 species in terms of their vegetation classification, their nectar spur inner surface, as well as their pollinator rewards. The study region was in the Northern and Western Capes, around Nieuwoudtville and Neverus, respectively. The image on the right shows the study sites around Nieuwoudtville. The study species were selected due to their morphological similarity, tendency to occur close together but not intermingling, and their spur differences and similarities. Nemesia nisacarpa and Palchella share similar lengths and downwards projecting orientation. Nemesia macroceras has the longest spur length of the 3, but also has an orientation of projecting forwards but deflects forwards. The Bromblunke phytosociological technique was based off of the guidelines stated by Brown et al. 2013, with considerations given by the studies. The data was captured in an unpublished macros-enabled Excel spreadsheet called VegCap, and then imported into JUICE, where a modified twin span classification was performed. The presence of nectar was observed on living individuals. In the nectar test, we measured the volume and sugar concentration of nectar by opening the mouth and inserting the microcapillary tubes into the nectar spur as indicated in the GIF. Measurements were taken from 10 flowers of each species. For the inner nectar spur surface observation, the samples collected in 3% glutaraldehyde were fixed in to fix any potential lipids. The nectar spur was dissected into dorsal and ventral, as well as into base, middle, and tip. The standard SAM preparation of samples was followed, with the samples being sputter-coated in iridium. The images were captured using the dual SAM at the Centre for Microscopy at the University of the Free State. Ruzin's 1999 book on plant microtechnique and microscopy was used as a guide for the microtechniques and sample preparation and staining procedures. The saffron O and orange G staining technique was based off of Sharman's 1943 procedure. The PAS staining protocol followed was that of the PAS staining kit by Sigma Ulrich, as well as Ruzin's PAS protocol, where the aldehyde blocking step was incorporated. The 3 Nemesia species are associated with 3 different vegetation communities. Nemesia nisocarpa is associated with Community 1, Nemesia paucella with Community 2, and Nemesia macroceras with Community 3. So Nemesia nisocarpa is found in the succulent shrub community Lampranthus watermeri, Stuberia frutescens, and the vegetation is mostly succulent dwarf shrubs, and herbs. The plants grow closely together in patches with bare soil in between. The herbs were often found growing beneath, within, or near the succulent or dwarf shrubs. Exposed bare soil is a prominent feature within the vegetation, and this community was found on a sandy, northeast-facing, gradual to steep foot slope of a hillside. One of the community's naming species, Stuberia frutescens, is a biogeographically important species of the Namakwaland Klipklopper shrubland vegetation unit of the Namakwaland endemics. This community can be loosely compared to the succulent dwarf sparse shrubland community of the Gughab Nature Reserve. The Nemesia paucella Hesperantha cuculata Ixia rapunculoides subcommunity is dominated by herbs and geophytes. The shrub layer is dominated by Muraltia spinosa, and the vegetation is the most prominent feature within the subcommunity. This subcommunity is found on loamy sand of a plain that had a flat slope. This subcommunity resembles the Nevoidville mosaic observed in Van der Merwe et al.'s 2008 study, as well as fragments of the Nevoidville Roggeveld Dollerite Renosterveld and the Nevoidville Shale Renosterveld vegetation units. The mosaic described by Van der Merwe et al. Consisted of 3 vegetation units, all of which are resembled within the Nemesia paucella subcommunities. The Nemesia paucella Hesperantha cuculata Nemesia leipolti subcommunity is dominated by herbs. This subcommunity showcases the most graminoid species amongst the subcommunities and communities surveyed within the study. The shrub layer is dominated by Eriocephalus punctulatus, and it is found on the sandy loam of a north to east facing flat to gradual foot slope of a hillside. There is no outcrop present within this subcommunity, and bare soil was very low. Nemesia leipolti was also found to be fairly common within this subcommunity. The Nemesia paucella Hesperantha cuculata Aerospermum capensa subcommunity is dominated by herbs and geophytes. The Diospyros austroafricana and Viborgia monoptera dominate the shrub layer. This subcommunity is found on a sandy southeast-facing plain, and it perfectly represents the Nemesia paucella population at the Papkelsfontein guest farm. Nemesia gracilis was found to be a rare community member. The subcommunities of Community 2 can be distinguished through the absence and presence of certain groups of species, and most noticeably by that of the dominant shrub species. Nemesia macroceras is associated with the Restioland Vildenoa incovata Osteospermum monstrosum community. Herbs and geophytes dominate the community, and plants in this community grow within patches of shallow soil between large outcrops. This shrub layer was well represented by the restioid species Vildenoa incurvata and shrub species Muraltia spinosa. The community is found on a sandy south to southeast facing plain, and the soil was shallow as outcrops were prominent within this community, with sizes ranging from gravel to boulders. This community perfectly represents the Nemesia macroceras population found at the Babkelsfontein guest farm, and it contains the biogeographically important and endemic taxa of the Bokkerveld sandstone fynbos. However, it is also possible that the vegetation is part of the Nevotville mosaic. Although Nemesia paucella and macroceras occur on the same farm, they occur in different vegetation communities and do not share pollinators, as indicated by their spur morphology. Here are the results of the nectar test. Nectar was observed in 2 out of the 10 flowers of Nemesia paucella, yielding a volume average of 0.042 microliters and an inconclusive sugar concentration. Nemesia macroceras had a nectar volume average
It's all right, Emma, take your time. No problem.
You could hear
No,
that.
no problem.
The Nemesia macroceras had a nectar volume average of 0.309 and the average sugar concentration 0.13 bricks. The result of the inner surface observation showed the presence of trichomes on the ventral basal surface of these species. It is important as trichomes are useful to a pollinator by increasing grip. This indicates that the ventral base surface of the nectar spur is expected to come into contact with pollinators. The ventral surface of Nemesia anisocarpa has dense trichomes that become less dense and shorter down the length of the spur, and the inner spur surfaces of Nemesia paucella are variable but distinguishable down the length of the spur. The variations in trichome shape and density are seen on both the ventral and dorsal surfaces, and this is the ventral variation. This is the dorsal variation. And interestingly, the dorsal surface shows the presence of trichomes instead of the ventral surface. This is the ventral and dorsal inner spur surface of the base section of Nemesia macroceras, where the ventral surface has sparsely placed linear trichomes. Due to the nectar spur length of Nemesia macroceras, there is a greater variation within the middle section. Here is the ventral and dorsal surfaces, where the ventral surface has trichomes and the dorsal doesn't. Lower down the nectar spur, the cuticular striations become smoother, and the point of deflection could be distinguished by the contraction of undulating epidermal cells. And due to the nectar spur length, there is greater variation. Okay, wait. Lower down the nectar spur, the cuticular striations become smoother, the point of deflection is seen. And here we are at the tip surfaces, and we can see the ventral and on the ventral surface, we can see the glandular trichomes. So this is the dorsal surface trichome arrangement, which has a unique U shape, which distinguishes it from the ventral surface. Here you can see the remnant of where the nectar used to accumulate. So the safranin O stains lignin, cutin, suberin, chitin chromosomes and nucleoli red, whilst orange G stains the cytoplasm yellow to orange. And Here in the base of Nemesia paucella, we can see the vascular tissue and trichomes are stained red to orange, with both indicate high concentration of nucleoli. In the middle section, the base of the trichomes can be seen as they stained red in between the epidermal cells. The trichomes in the base of Nemesia macroceras did not stain as brilliantly red as those seen in Anisocarpa and paucella. The glandular trichome seen in the tip section stained a brilliant red to orange, and it can be compared to the clavate trichomes of Nemesia anisocarpa and paucella. The pass reaction showed that the trichomes of Nemesia anisocarpa are hollow with an outer and inner tube. The base and middle section showed the presence of starch within the trichomes. And the trichomes and papillae observed in the middle section have an outer and inner layer to the trichome, and the inside contains a secretion that consists of polysaccharides. Here I've included the tip section of Nemesia macroceras, and they showed both empty and full glandular trichomes, with the secretions image on the far right proving to be rich in polysaccharides. As PAS reacts to polysaccharides and turns them purple. In addition, the secretion seen in the right image is clearly nectar, as the osmium tetraoxide fixation did not stain any of the molecules black. Okay, so in conclusion, the three species don't have the same plant-plant interactions and don't grow together. Nemesia anisocarpa is associated with the succulent dwarf shrub community, Nemesia paucella with the vegetation mosaic described by Van der Merwe et al., and macroceras with the restio shrubland. The vegetation associated with the Nemesia species is indicative of environmental or edaphic preferences and provides a baseline for further ecological studies within the genus. The presence of trichomes in the basal ventral inner nectar spur surface indicates the pollinator interactions with the nectar spur, and one out of the three species conclusively produces nectar as a pollinator reward, and Nemesia paucella has the potential to produce nectar but perhaps not secrete it, and the nectar spur morphology is indicative of nectar production. These are my acknowledgements, my references. Thank you.
Thank you, Emma. Very, very interesting. It's great to see also the use of vegetation classification. There's one question. There's not a name add to it. Thank you so much, Emma. Great talk. You mentioned that the morphological differences between species could infer that they do not share pollinators. Did you notice different pollinators in the field?
Indeed, we did. Well, for the one species, at least for Nemesia paucella, we observed a bee collecting pollen off of the out of the becky, and we did not observe that pollinator for macroceras, but due to the spur length and the Darwin theory, we can assume that there should be a different pollinator. I hope that answers your question.
I do think so. Are there any other questions? You can just post them on the Q&A box. Or if you want to ask a question directly, you're welcome to just switch on your mic and your cam. Okay, seems to me that there are no more questions. Thank you so much, Emma, for a very interesting talk. I hope you are well. I hope you are okay. maybe drinking too much water. But
I was diagnosed with COVID, so it's a good thing we are doing this virtually.
Oh, wow. Yeah. Well, good luck for you then. Okay. Thank you very much.
Thank you. Bye.
All right. That brings us to the third presentation in this session. It is by Konrad Geldnys, and he will tell us more about a new conservation area of the Guchap Nature Reserve. Over to you, Conrad.
Thank you very much. I hope everybody can see my screen. So I'm going to talk about a new conservation area adjacent to Guchap Nature Reserve just outside Springbok in the succulent Karoo, and specifically focus on the monitoring that we've been doing there over the last 11 years and what we found. And it coincided with a period of severe drought. So I will give brief background of the project and the literature around monitoring, and then also discuss the study area, the methodology that we used, and then the brief results and summary. So the aim is to establish a baseline from which to assess this change from the previous livestock agricultural land use to the present conservation land use, and also to inform Guchap Nature Reserve's rangeland and wildlife management decision-making, which is in line with the management plan and the conservation objectives of the reserve. But there's this byproduct also, or additional benefit, that we can monitor long-term changes in species composition and vegetation structure over time by doing these surveys annually. There are various studies looking at long-term changes in vegetation in the succulent Karoo, and there are different approaches. For example, Nenzalele and co-authors, they looked at decadal changes at 10-year interval surveys, looking at changes between commercial and communal properties on a fence line. And then there's Hoffman et al.'s approach, where they use NDVI changes in the long term, as well as repeat photography, historical photograph records to look at changes. And then there's the other type of surveys like Van Rooyen et al. And Ute Schmiedel's group, where they do annual surveys in the field every year and look at changes that occur locally. And for interest, the Van Rooyen study is a study that's been going on for more than 40 years on Guchap Nature Reserve. So the study area is located in the succulent Karoo. It's indicated here in the purplish colors, the different vegetation units that you find there. And in yellow is the Nama Karoo, the Bushmanland. So it's directly located on this ecotone area. So the reserve was established in 1966 and gradually expanded. And then since 2009, a number of additional properties were added to the reserve, indicated here in green, bought by the WWF. And then there's also a biodiversity offset property in green. And as you go further east, there's livestock properties, and an additional conservation area for interest was also established recently at the Karasbergen Nature Reserve around these Inselbergs over here. If you look at the climate of the area, specifically focusing on the annual rainfall, now this is the Guchap Nature Reserve data record for rainfall on the reserve. And you can see that since 1973, in this case, there's very distinct rainfall cycles of wetter periods and dry periods and something in between. But you will notice that from 2015, right at the end, for 7 years consecutively, there was below average rainfall, below the long-term average of around 150 millimeters. So this, I think one can safely say, was a period of a severe drought. If we look at the rainfall seasonality, on the left you have Guchap Nature Reserve's data. So you'll see there's a clear indication that this is winter rainfall dominated. From May to August is the main rainfall season. And if you go further east, this is the rainfall data for Arep livestock farm. You'll see that the rainfall is more spread out throughout the year, the stronger contribution from summer rainfall, and the quantum also becomes less as you go further east. But for the purpose of this study and this presentation, we will focus on this specific property, Rafelkraal South, where the yellow star is, and that is very much a winter rainfall dominated area. So for the methodology, annual surveys, field surveys are week of August or first week of September, using line transects of 500 meters per site. And we use the descending point method of Roux. So you have 500 points, data points per site. And at each meter interval along this line transect, you record the first plant that you strike with a descending rod, as well as the second plant that you strike, since some plants are than others and they may hide under the canopies. We calculated range condition using the grazing index method and also looked at the change in Shannon-Wiener's index of diversity. And we also looked at how growth form abundance changed. Here we focus on the frutescent gametophytes, for example, the Karoo bossies like Eriocephalus, the semi-woody ones like the subfrutescent gametophytes. And then there's also the leaf succulent gametophytes, which is your mesem shrubs mostly, like Russia robusta. And then also the hemicryptophytes, the perennial grasses, and the pterophytes or the annuals. So over the years, a large number of permanent vegetation monitoring transects has been established over this whole conservation area, over the Greater Kruger Conservation Area. Excuse me. And the first ones were started in 1974 on the reserve itself. Since 2011, we started to put out transects on this extended conservation area as they were added. And for this presentation, as I indicated, we will look at 4 transects on the Radelkraal South property, and we will look at 2 transects in the Hills habitats, in the Hill valleys, and then also 2 transects on the sandy plains towards the right. So this is what it looks like. There are 2 habitats on the left, 2 monitoring sites, which is in the mountain valleys. It's generally sandy vegetation, but there's a larger component of gravel and rock present than you will find in the sandy plains, which is exclusively consists of sand. If we look at the rainfall on Radelkraal South specifically over this period from 2011, indicated by the green arrow, you will see that initially at the start, there was fairly good rainfall, variable, but not too bad. But from around 2015, there was a sustained period of below average rainfall, both in terms of annual rainfall, indicated in red, and winter rainfall also, as indicated in blue. So if we look at what happened to rangeland condition change over this period, as calculated with the veld condition score, you'll see that there was a significant decline in rangeland condition in all the habitats up to present. If you look at the slope of the plains habitats, which is in green and blue, you'll see that the slope is much sharper or slightly sharper, indicating a faster decline than the Hills habitats, but all of them declined nonetheless. And for interest, if you look at the veld condition score, you'll see that some of the habitats are approaching 0, indicating that there is very little live perennial matter left. At the start of the monitoring period, game numbers or pressure from game grazing could have played an additional factor. So if you look at the game numbers for the entire Kruger Nature Reserve, so this is not just for Radelkraal because we can't separate them, it's an open system. You'll see that from 2011 to 2014, if you look at the black line, the Gemsbok numbers were initially elevated. But as the veld condition deteriorated and the drought started to set in, drastic measures were taken to reduce the populations of the game, especially the Gemsbok. So you'll see that the trend indicates that there's been a sharp decline in the Gemsbok numbers from around 2014-15, as well as that of Springbok, indicated by the green line, and also the ostrich in pink, but that was not actively culling, that was just natural die-off. It's only the Hartmann's mountain zebra numbers indicated by the red line that will remain constant. So what happened to plant cover if we look at the trend? If we first look at the black lines, that indicates what happened to total vegetation cover in all of the habitats. There was a decline but it was only significant in one of the habitats, one of the mountain valley habitats in terms of habitat B. But if we look at perennial vegetation cover, then in all habitats, there was a significant decline over the monitoring period. And now, if we look at the perennial life form cover, which I showed earlier, if we focus first on the left and look at the mountain valleys, what happened there? So in black is indicated the fruitescent chametophytes, or the karoo bossies. You can see that they declined significantly, as did in red the leaf succulent chametophytes, which are the mesum shrubs. And in green is indicated the semi-woody chametophytes, so semi-woody shrubs. And they did decline, but it was not significant in one of the habitats. If we move to the right and look at what happened to the sandy plains, here the hemicryptophytes or perennial grasses is a more dominant feature of these habitats. And in the top graph, it is indicated in green, and in the bottom graph, it's indicated in blue. And you can see in both instances, the decline was significant. And also the karoo, the mesum shrubs in habitat A, the black line, declined significantly in habitat A, and also the woody shrubs or the karoo bossies declined significantly in habitat B as well. What happened to annual lifeform cover? Now, the annual lifeform cover is very much dependent on the amount of rainfall that is received, especially winter rainfall, since this is a winter rainfall region. So you can see on the graphs, if you just look superficially, you will see that as rainfall increases, indicated by the blue lines, then the annual cover is generally higher, indicated by the black dots. Over time, though, it shows that at least in the mountain valleys habitats, there was a significant increase in annual cover over time. But this may be an anomaly based on spatial and temporal variation in rainfall, because it can be different in closely situated areas. But also, as the perennial cover declined in time, there's more opportunity for the surveyor to strike annual cover plants or annual species that are in between the shrubs. So it may be actually a function of what we observe here. Species richness. So total species richness did not change significantly over time, but if we look at perennial species richness in all 4 of the habitats, there was a significant decline over time. And then if we look at Shannon-Wiener Index of Diversity for perennial species specifically, there was no significant observation of change over time. It was very flat, except for one of the sandy plains habitats where there was a significant decline in time. These tables show the relationship between rangeland condition and the different community variables that I just showed, just to show what are the important indicators that seem to correlate with a reduction or an increase. And the common denominators here generally are perennial species richness, the total cover, perennial cover, annual rainfall, and then to a lesser degree, the Shannon-Wiener Index diversity. So in summary, this 7-year drought period has caused a significant deterioration in rangeland condition in both the plains and the hills habitats. And the pressure on the rangeland from grazing initially at the onset of this survey period may have provided an additional impact on the change in rangeland condition going further into the drought period. Perennial cover and species richness declined significantly, as indicated. Total cover declined significantly in the hills, but in the plains, the decline was not significant. And then species diversity was only correlated with decreasing rangeland condition in one of the habitats. And then, as I mentioned, there increased significantly with rangeland deterioration in one of these hills habitats especially, but there may be other factors explaining that observation. I would like to thank all my colleagues who assisted over the years with the surveys and still continue to do so, hopefully in the years to come. Our thanks to the WWF and the Leslie Hill Succulent Karoo Trust for funding for doing these surveys, and also for co-researchers in assisting with reviewing this presentation. Thank you very much.
I'm sorry, Conrad, I was disconnected. I suppose you've completed your I was disconnected for a while. There are some questions. Thank you very much for a very interesting contribution. Peter Malan also agrees. It's a very interesting talk. He asked if there are any sign yet of rangeland condition changes during COVID and also pre-COVID times. Have you witnessed anything? Have you looked at that?
COVID, I don't think it really. No, I can't imagine that. I don't think there's any relationship between what has happened with COVID and our societies, as you say, and what has happened to the rangeland condition change. I don't think there's any connection there.
All right. So, in other words, there are no humans. The local people have been moved away, so they are not using that area. And I just would like to ask, in terms of livestock grazing, is there any livestock grazing going on there?
No.
Apart
So
from
on the
specific game, nothing.
Yeah, no. So the area that I discussed also now that Rattlekraal South property, it used to be a livestock farm up to about 2009 when it was incorporated into the reserve. And prior to that, the landowner had stopped farming for, I think, 5 or 10 years. So effectively, there was no grazing really. So I think, and if you look at the historical rainfall records, the property was at that stage in probably a fairly good condition prior to what has transpired now with the drought.
Okay. Ladies and gentlemen, are there any more questions to Conrad? You can post them or you can ask them yourselves. Thank you very much, Conrad, for a very interesting talk. Apologies that I was not present during the conclusion part. Thank you very much. All right, I would like to introduce the fourth presentation, the last one before our break. It is Nanamshla Gwedla that will tell us more about health clinic garden research in South Africa. Thank you, Nana. Over to you.
Just to be sure, can everyone see my screen?
It's fine, you can proceed.
Okay. Good afternoon.
Sorry, okay, got in full presentation. Thank you, sorry.
Good afternoon, ladies and gentlemen. My name is Nanamshla Gwedla, and today I'll be sharing with you results from one of the studies from the broader Health Clinic Gardens research that is entitled South African Health Clinic Gardens as Models for Social Ecological Systems, and we're looking at plant diversity and potential utilization. Gardens as essential types of green infrastructure are the most interconnected urban and rural areas and have a great influence on human health and well-being, because of their close connection to humans. Gardens can be classified into different forms of ownership with the purpose of determining the variety of ecosystem services that they provide. Now, types of gardens can include those that are privately owned, and here we're talking about domestic gardens, home gardens, or even community gardens. Then we have those that are entity-owned or government-owned. Some are funded by the government and managed by the government, and these include botanical gardens and school gardens. In South Africa, health clinic gardens are another type of garden that is developed by different stakeholders on government-owned land. Now, in line with the provision of ecosystem services for improved quality of life, different types of gardens grow different plants that provide varying utility, that provide a variety of utilitarian properties. And these include types of food and medicines, but these should not just be focusing on food and medicine, but equally be considered for their provision of other ecosystem services as well. Plant selection decisions in gardens is determined or is driven by several factors, including socioeconomic factors, environmental priorities, and geographical regions, as well as owner preferences, because ultimately the aim is to enhance ecosystem services provision. Now, a few studies have explored plant distribution, plant diversity, and utilization in various types of urban, peri-urban, and rural gardens across South Africa. And most available studies focus on different gardens, such as home gardens and community gardens, as spaces for food security and the cultivation of medicinal plants. Some of the detailed studies on the distribution and potential uses of plant species in gardens include that of Nemu-pukke and colleagues in 2010, who found that rural home gardens around KZN have a higher percentage of naturally occurring indigenous useful plants compared to peri-urban areas. Lieb and others in 2011 later assessed the floristic composition of domestic gardens in Tlokwe, and they outlined the contribution of domestic gardens to the plant diversity of urban ecosystems. A seminal contribution on distribution, diversity, and utilization is perhaps that of Malebatsi and others in 2010, who investigated the different use categories of garden plants in the Tshwana Chimo garden layout and found over 500 useful plant species from 105 different plant families. Now, bringing this back home and illuminating the social ecological nature of health clinic gardens, Celia et al. 2018 recently explored garden ecosystem services in sub-Saharan Africa and the role of health clinic gardens as social ecological systems, and asserted that health clinics provide ideal green infrastructure because they are government funded, they are maintained by the government, and they are linked to free healthcare and stand central within communities, making their gardens ideal social ecological systems for future studies towards social ecological innovation. Now, the model of health clinic gardens is prevalent in South Africa, and their high visibility and accessibility positions them as sustainable gardens and strong cases for usefulness for other countries to follow. Against this background that I've just given you, this study sought to provide insight into the plant species composition of health clinic gardens to establish and ascertain their potential uses and position these gardens as alternative models to gardening in rural, peri-urban, and urban areas. This study was conducted in the Northwest province of South Africa, which is, and the specific study area was the Bojanala district across 5 local municipalities. This district is in the savanna biome, central bushveld, and is characterized mainly by thorny woodland vegetation. The province has just over 500,000 households, and it is extremely high in poverty. Platinum mining, industrial development, as well as recreation are major economic drivers in the province. Now, how did we do this study? Very briefly, across 103 health clinic gardens, we were looking at peri-urban and rural areas mostly, using the Tshwana Chimo garden layout, and we were identifying plant species This happened in spring 2015, and we were classifying plants into different categories. And we were using different instruments such as the frequency of occurrence. We use literature sources to identify some of the plants, as well as researcher observations to also look into how these plant species may be used, or just to observe them and see what kind of species they are. And in terms of analysis, we were just, for example, enumerating the number of gardens that have a specific species. And we were using basic statistics for correlations and differences in species composition. And the ANOVA tests were mainly used when we had to do statistical tests. Now, what did we find out? 116 plant families were found across all health clinic gardens, and these comprised of 404 genera and 645 species. And some of the most frequently encountered families included the Asteraceae, Poaceae, and Fabaceae. And among the recorded genera, those species that were previously included under Vechelia and Senegalia, as well as Eregratis and Euphorbia, were the most frequently occurring. They consisted mostly of indigenous species also, and there were some that were native to the Northwest Province, while those that were in the Euphorbia genus were mostly exotic. In terms of the most frequent species, we found grass species, which is the Cyodon dactylion as the most frequently occurring. And these are both native to the Northwest Province. And finally, herbaceous plants were the most dominant, followed by trees and shrubs. When we look at the life forms, phanerophytes were the most abundant, and these were represented by 195 species. When we look at the origins of these species, 321 out of the 645 species that were recorded, they were indigenous to South Africa, and of those, 210 were native to the Northwest Province. When we look into the differences in species assemblages in health clinic gardens between the different local municipalities, these yielded no significant differences. So there was no significant variation here. While there were significant differences in the number of species that were found between the various microgardens within the health clinic gardens. And here we found that the Naga microgarden had more species than other microgardens, as we can see in figure 3 on the right. Taking a look at the classifications of plant species, we found that 14 of the exotic species that were encountered have been declared as invasive in various categories. As we can see in the table, we've got C3 invaders, category 1B invaders, and category 3 invaders, for example. And these originate from various parts of the world, such as Mexico, Madagascar, South America, Central, Eastern, and Northern China, just to name a few. Now, despite their invasive potential, many of these have been found to be useful, as we can see in the last column, where some are used for food, such as fruit, medicinal, ornamental, etc. Looking at the second key finding, many of the encountered species across all the gardens have been identified as potentially useful. And here we're looking at about 426 species. And they have various purposes, including food and medicine. And some of these species overlap in terms of their uses, where one can be used both as a medicine or as a type of food or a fruit. An example of this could be the Ziziphus mucronata, which is used both medicinally and as food. And the most dominant potential uses were the ornamental, medicinal, and food uses. And the ornamentally useful species were topping this list. We can see on figure 4, this ornamental species were just close to 200 compared to species that were useful as a hedge, which was pretty much less than 10, so to speak. Now, figure 5 illustrates the distribution of potential uses among those categories that I've just mentioned. And we deliberately picked out the medicinal and the food ecosystem services here. And if we look at in the first part, which is the potential medicinal uses, we can see that plants that are used as a tonic were the most abundant, followed by those that are used to treat general ailments, and we also categorized those as general medicine. On the right, where we're looking at the foods that are species that are useful as food, we can see that fruit species were the most abundant, followed by leafy vegetables, as well as African leafy vegetables. In terms of the specific plants that are potentially useful, the Agapanthus species, for example, was one that was encountered the most. It was found in 40 gardens in the category of medicinally useful plants. And it's an indigenous plant, which is a good thing. And unfortunately, it is poisonous, just as an example. And when we look at the edible plants, we're looking at the grass species, which is the Urocloa mosambicensis, and this can be used as a grain. Fortunately, again, this one is native and it was found in 83 health clinic gardens. Now, drawing this back and making conclusions, we can see that much like home and domestic gardens and other type of gardens that have been investigated in the past, health clinic gardens have a variety of plant species that represent an enormous number of genera, as well as coming from various families. We also see that plant resources support humans and provide goods like food. And here we're talking about fruit, we're talking about grains, and we're talking about vegetables. And there are also medicinal plants. In other instances, we see fodder as well. We need to admit that plants also provide cultural services such as spiritual, aesthetic, and inspirational values. And a very clear example of this was the presence of hedges in some of these health clinic gardens, which for some people may serve as an aesthetic value. As a result, given all these supporting services to humans or provisioning, we can conclude that gathering activity for what many may term as wild plants is rapidly increasing. That is understandable because people seem to be using these plants, or to put it in another way, most of these plants seem to be useful to people. Now, health clinic gardens in the Northwest are very much reminiscent of other garden types, and they reflect most of the basic structure of other garden types, with very minor differences here and there. Therefore, I think it's safe to say that health clinic gardens around clinics or hospitals can be used as an alternative to other types of privately owned or entity or even government-owned established gardens, where challenges such as lack of land, for example, lack of security, shortage of water, they continue to persist. Now, given the envisaged purpose of health clinics, future research, health clinics and health clinic gardens, future research could investigate how traditional knowledge on useful indigenous and naturalized species can be illuminated and restored to position these gardens or health clinic as spaces that will encourage their use as well as champion their supply among communities, especially for poor households. And
you've
these
got
are the
1 minute left.
These are the references that we used in the study. And I'd like to acknowledge facilities managers as well as groundsmen, the NRF for funding the study, as well as the NWU for funding the study, and the cooperation from the health department, the provincial health department. Thank you very much.
Thank you, Nana. There you are. Thank you, Nana. Ladies and gentlemen, are there any questions? Maybe just a question from my side, Nana, in terms of further use of the data that you've presented here. How would you go about to use that specific information in future studies? In terms also of the social aspects that, as you mentioned in the beginning.
Okay, the immediate thing that jumps up to me was that, for example, this data that was collected for this particular presentation that I've given today, it wasn't, for example, verified with the communities or the people who are supposedly using these plants. So one way that could that this kind of data could be used socially would be to, for example, compile a list of these species which we've identified as potentially useful, and actually go into the communities, the people who are supposedly using these species, and actually find out from them use them and how maybe they can I don't know, give pointers on how such species can be conserved or preserved. And I think the keynote address in the morning did speak a little bit to something like that. I hope I'm answering your question.
Thank you very much. Are there any questions? All right, thank you, Nana. That wrapped this first part of our session 1 up. Thank you very much. I would like to thank all the presenters and all the people who also posed questions in the audience. We will have a 10-minute comfort break now, and then after that we will complete with 4 more presentations. Thank you very much.